So we begin our class. In last class, we have discussed about the etiological characteristic of staphylococcal infections, which is caused by a bacteria. That bacteria we call it as a Staphylococcus aureus, and this bacteria has a characteristic that it produces coagulase enzymes. It produces coagulase enzyme, which is helpful for this bacteria to invade the host and cause the infection. So the next point in this staphylococcal infection is the epidemiology of this disease. Now the is Staphylococcus aureus or staphylococci. They are the primary parasites of human beings and animals. They are mostly colonizing the skin glands, and mucous membranes. The most common source of this infection for The most common sources of this staphylococcal infection are the human patients and the carriers. So the source for this staphylococcal infection, they are the patients and the carriers, but mostly they are the human patients and carriers. Animals and inanimate objects, they are also less important, but they can also initiate the staphylococcal infections. Now the patients with superficial lesions and respiratory infections, they give out large quantity of this staphylococci into the environment. About 10 to 13% of the healthy person, they carry the staphylococci in their nose, and about 10% they carry this staphylococci in perineum and also on the hairs. The vaginal carriage is also about 5 to 10%, which rises greatly during the men's. A factor relevant which now this presence of this bacteria in vaginal carriage, this is responsible for causing another dangerous disease in women, that is toxic shock syndrome, that we are also going to see in the next point. Now staphylococcal carriage, they starts in early in the life. Colonization of umbilical stump being the very common in babies which are born in hospitals. Some carriers can give out this bacteria, so they are also called as shedders because they disseminate large quantity of this cocci for longer period. So because of that, these carriers, they are called as shedders. They give out this bacteria in large quantity for longer period. Now these cocci, they can be given out by the patients and carriers which contaminate the fomitids such as handkerchiefs, bedlines, blankets, and they may persist there for days or weeks. So this bacteria, when they present on these fomitids, they can also act as a source for healthy persons. Now this staphylococci may also come from infected domestic animals such as cow. If cow get infected with staphylococcal infection, from that infection can be transferred to the healthy human beings. Now the staphylococcal disease, they may be endogenous or exogenous infections. When sometimes the bacteria which is present on the skin of that healthy person, they may initiate the infection in that, so it becomes endogenous. And exogenous means from other source. Sometimes the infection can be initiated in that host. So this mode is called as an exogenous infection. Now the mode of transmission of this infection is by contact, direct or through the fomitids. It may be by dust or maybe by airborne droplets. So the sources are the human patients and carrier. The mode of transmission, they are the contact, direct or by through the vomitus, or by dust particles, or by airborne droplets. So this plays a very important role in order to develop this staphylococcal infection. What plays a role? Patient, human patients and the carrier, they are the sources, but the mode of transmission, it is the direct contact or through the vomitus or by dust. Now, this bacteria, it can be also cause infection in hospitals. Now, special attention should be given to those infections which are caused in hospital because of this staphylococcus. There is a reason behind this, because the most of the strains which are present causing this infection in hospitals, they are resistant to various antibiotics. So this becomes serious one, because if no antibiotic can kill this infection, this pathogen in the hospital area, because due to the use of various kinds of antibiotic in hospital areas, this bacteria also become resistant to various antibiotics. Now this staphylococcus, they are also causing common post-operative wound infections and other hospital cross infections, and most of this due to the certain strains of staphylococci that are present in hospital environment. Those strains which are most commonly found in the hospital environment and causing such type of post-operative wound infection and other hospital cross infections, such strains, they are called as a hospital strains, and they should be taken care of because of their ability to resist the various kinds of antibiotics. Now, here these strains, hospital strains, they can be typed. Fast typing is carried out, and they are most commonly resistant. The hospital strains, they are most commonly resistant to penicillin and other antibiotics which are routinely used in hospitals. So some of them, they are the epidemic strains causing epidemics in hospital cross infections. So they are also taken care of because they are having a resistance characteristic against the common antibiotics which are used in hospitals. So such strains, they can cause epidemics in the hospital area by causing hospital cross infection, and these strains, they are also called as epidemic strains because they cause epidemics in hospitals. Now the first of these to be recognized was of fast type 80781. This is a strain, Staphylococcus strain, which is accounted for the most of the staphylococcal infections in hospitals throughout the world. This is a Staphylococcus strain which is having fast typing 80781, and this strain is most commonly causing hospital infections throughout the world. जिथे जिथे हॉस्पिटल्स आहेत त्या हॉस्पिटल्समध्ये ह्या स्ट्रेन जो फास्ट टाईपने केलेला आहे त्या स्ट्रेननं स्टॅफिलोकोकल इन्फेक्शन्स हे मोठ्या प्रमाणामध्ये कॉज केलेले आहे. सो दिस स्ट्रेन दे हॅव बीन since replaced by other strains of Staphylococci and other gram-negative bacilli. So there may be change occurred because of the development of antibiotic resistance in this group of bacteria. Now what measures should be taken in order to avoid or control the staphylococcal infection in hospitals? First precaution which is taken is the isolation of the patients with open staphylococcal infections. Such patients which are having staphylococcal infection, they should be isolated from the other patients. Secondly, detection of staphylococcal lesions among the surgeons, nurses, and other hospital top staff. Is also an important point because staphylococcal infection may be transferred from the surgeons, nurses, or other hospital staffs which are carrying this. So that should be detected, and they should be kept away from the department whenever that person may heal from that infection. Then third, strict aseptic techniques in operation theaters. Fourth, oldest and simplest and the most effective method of checking hospital cross infection is the hand washing. But this hand washing technique, it has been neglected. If an outbreak of Staphylococcus sepsis occurs, search may be carried out in order to find out if some type of carrier may be there in the hospital staff. Okay, if Staphylococcus sepsis occurs, the search should be made for the carrier in order to detect the carriers in the among the hospital staff, and those detected should be treated with local application of neomycin and chlorhexidine. These are the antibiotics which are used for the treatment of those persons which are carriers. Now, in some institutions in America, the eradication of the virulent strain, resident strain, has been carried out by using a strain which are having low virulence power. This low virulence power strain, it is introduced in the hospital area, so that because of this, the strain which is the highly virulent in the hospital area, this is replaced by this low virulent strain. And antimicrobial prophylaxis by topical application of antiseptics such as hexachloroquine has also been found useful in hospital areas. So these precautions should be taken in order to avoid hospital cross infections because of the Staphylococcus aureus. Then the next point is the pathogenesis and the clinical features of this staphylococcal infection. This staphylococci produces two types of diseases. First is the infection and the second is the intoxication, means food poisoning. In Staphylococcus infections, the cocci gain access to damaged skin, mucosal and or tissue sites. It colonizes there by adhering to the cell or extracellular matrix, innervate the host defense mechanism, multiply and cause tissue damage. So such type of infection we called as a staphylococcal infection. Now in intoxication, the disease is caused by bacterial toxin, which is produced either in the infected host or preformed in vitro. If contaminated food by this pathogen where it produces toxin, if such toxin contaminated food is eaten by some healthy person, the food poisoning may occur or food intoxication may occur in that person. Now this staphylococci causes diseases which includes acne, boils, pimples, impetigo, pneumonia, osteomyelitis, carditis, meningitis, arthritis. These are the most common infections, staphylococcal infections, which is caused by the Staphylococcus aureus strain. And many of these diseases, they are pyogenic, means they forming the pus. Okay, those diseases which are causing pus, these diseases are known as the pyogenic diseases. Now, healthy individuals, healthy individuals are often carriers, and resident staphylococci in the upper respiratory tract or skin seldom cause diseases. Means in healthy person, this Staphylococcus aureus is mostly found there, and in some condition it may initiate the infection. It also present in the upper respiratory tract or also present on the skin. So they may initiate the infections. Now during the first week of the life, infant often become infected by this pathogen either because of the mother or from any other close human contact. And serious staphylococcal infection can often occur when the resistance power of the host is low. If the infant in the first week, if it comes in close contact with other humans which is having these infections caused by the staphylococci. If mother is suffering from this infection, then such infection may be transferred to that infant, and whenever the immune power of that infant becomes low, at that time this staphylococcus may cause the infection. And how this low resistance power becomes low? It's because of the hormonal changes, debilitating illness, wounds, treatment with steroids, other drugs, and because of this the immunity gets lower, and in such conditions the Staphylococcus aureus may initiate the infection in that infant. Now the Staphylococcus produces disease through their ability to multiply and spread widely in the tissues, and also by producing some many virulence factors. Some of these factors, they are the exotoxins. Some of them are the enzymes which help this bacteria to spread in the host tissues. Okay, now which are these? Here this bacteria produces 4 different type of hemolysin, which is the compound which is secreted by this bacteria, and this hemolysin can cause lysis of the RBC cells. Okay, in earlier etiological characteristics we have seen that the Staphylococcus aureus it is producing beta type of hemolysis. So this hemolysis is caused by this hemolysin, which is some kind of toxic compound which is secreted by this Staphylococcus aureus, and because of that the RBC cells they get lysed. Now another virulent factor which is produced by the Staphylococcus aureus is the coagulase enzyme, an enzyme which converts fibrin into fibrinogen and forming a localized clots. And the production of coagulase is generally associated with the pathogenicity of the Staphylococcus aureus. Now clotting induced by this coagulase results in the accumulation of fibrin around the bacterial cell. Now this production of this coagulase enzyme favors the growth of this, favors the pathogen. How? Because of the accumulation of fibrin around the bacterial cell wall, our defense system is not able to make contact with the bacteria and causing the phagocytosis. So because of this accumulation of fibrinogen around the bacterial cells, our phagocytic cells, they are not capable of engulfing this pathogen and killing. Okay, so it contributes to the pathogenicity of that Staphylococcus aureus. Now Staphylococcus aureus strains also produces leukocidin. It is a protein which destroys leukocyte cells, means WBC cells, and production of leukocidin in skin lesions such as boils and pimples results in host cell destruction, and is one of the factors responsible for producing the pus in these pimples or boils. Now, some strains of the Staphylococcus aureus are able to produce some proteolytic enzymes. Some proteolytic enzymes such as hyaluronidase, fibrinolysin, lipase, ribonucleases, deoxyribonuclease. These are the enzymes which are produced by this bacteria and these enzymes they contribute for the invasiveness of this staphylococcal infections which is caused by Staphylococcus aureus. Now the classical example of staphylococcal lesion is the localized abscesses. Now this localized abscesses mainly occurs on the skin, where this bacteria Staphylococcus aureus become established in the hair follicles, causing tissue necrosis, and which results in and also this bacteria produces coagulase there, which forms a fibrin wall around the lesion, which limits the spread of this infection. Now within the center of the lesion, liquefaction of necrosis tissue occurs, and the abscess spreads in the direction of least resistance. So in that localized abscesses where this pus formation takes place because of this various virulent factors which are produced by this bacteria, and this from this part, these lesions, abscesses can spread to those parts where the resistance power is low. Now, these abscesses may be either furuncles or carbuncles. These are the two types of abscesses which is produced by this bacteria. You can see this in earlier figure. Now, the central necrotic tissue drains and healing also takes place. This diseases, they can also heal after some time. Okay, it is not compulsory that this remain for longer duration. Now, this bacteria may spread from any focus to lymphatics and bloodstream to other part of the body. If this bacteria enters to the lymphatic system or blood system, from that it can spread to the other parts of the body. Now, in case of newborn infants, we have seen that impetigo contagiosa. It is a disease which is caused in newborn infants and children, and this is the superficial skin infection, which is showing a encrusted pustule, and this disease is known as a impetigo contagiosa, which is caused by the bacteria Staphylococcus aureus. And why this is in because of that there is change in color of the skin, and contagiosa because this disease can be easily transferred, or it can be easily spread through one person to other, like in nursery, in schools. So in that it can be spread through the contacts. So that's why it is known as a contagiosa. It usually occurs, this impetigo contagiosa, it usually occurs in the areas where sanitation and personal hygiene is of poor quality. Now, certain strains of Staphylococcus aureus, they are also responsible for causing toxic shock syndrome, a serious outcome of staphylococcal infection, which is characterized by high fever, rashes, vomiting, diarrhea, and ultimately the death of that person. Here, toxic shock syndrome was first recognized in women and was associated with use of highly absorbent tampons. In menstruating females, blood and mucus in vagina can become colonized by this Staphylococcus aureus, which is present on the skin, and the presence of a tampons concentrate this material and creating ideal microbial growth conditions. Tampons, they are one kind of it is like a sanitary napkins which is used in MC. So this is because of this bacteria, there is occur of toxic shock syndrome in women where the sanitation conditions, they are not good. Largely due to the education and alteration in the materials used in tampons, toxic shock syndrome due to the tampons use is now relatively very rare. Over 70% of the toxic shock syndrome cases results in death. So this much amount is very, very high, and it occurs because of this bacterial Staphylococcus aureus. Now, this toxic shock syndrome is also seen in men and women, and usually initiated by staphylococcal infection following the surgeries. Surgery झाल्यानंतर हे infection होऊ शकते. Now, this toxic shock syndrome, it is caused of a exotoxin which is produced by this bacteria, and this exotoxin is called as a toxic shock syndrome toxin 1, TSST1. It is a supra-antigen released by growing Staphylococcus aureus, and this toxin recruits large number of T cells and causing inflammatory response characteristic of supra-antigen reactions. So this TSST toxin, it recruits some large number of T cells there where this toxin is produced by this bacteria, and because of that inflammatory response developed there. And this TSS may also caused by supraantigens from other pathogenic bacteria such as Streptococcus pyogenes. Now another kind of infection which is caused by the Staphylococcus is the staphylococcal scalded skin syndrome. Is also an example, common example, which is caused by staphylococcal disease. This staphylococcal scalded skin syndrome, caused by the strains of Staphylococcus aureus, produces a hexafoliative toxin or hexafoliatin. It is a toxin. This protein is usually produced by plasmids, which is present in that bacteria. Some strains may carry the toxin production genes on their chromosomes. So it is a plasmid coded toxin or protein which is causing this type of syndrome. Now in this disease, the epidermis of that person where this toxin is produced is peeled off and reveal a red area underneath. That's why the name is given to this staphylococcal scalded skin syndrome, and is seen in most commonly in infants and children's neonatal nurseries. Now this yes is most commonly observed in infants and children. They are also found in neonatal nurseries. Occasionally suffers large outbreaks of this disease. This is the infections which is caused by the Staphylococcus. Here, first tissues where the Staphylococcus aureus is often found doesn't normally cause disease. These are the locations, and disease that may be caused by Staphylococcus aureus are pimples, impetigo, here on the skin of the chest, wound infections and abscesses spread to the lymph nodes and to the blood, septicemias resulting in widespread shedding, osteomyelitis, means they are causing infections on the bones also, endocarditis, here on the cells of the heart, meningitis, enteritis, enterotoxin poisoning, food poisoning, nephritis, in respiratory infections, pharyngitis, laryngitis, bronchitis, pneumonia. These are the various infections which are caused by the Staphylococcus aureus in a human being. So you can observe here. This is the superficial folliculitis, which occurs in those parts where hair is there. At the hair follicles, this syndrome, this type of lesions have been developed. Same is there. Deep folliculitis is there. Then furuncles is this. This one is the furuncle. Then a carbuncles is this one, which is present behind the neck of that person. Then impetigo. This is the impetigo, and this is a contagious disease. Any healthy person which comes in contact with such case, such infection may be transferred to that person also. This is another scaled skin syndrome, where the skin of that child is given out because of the production of this specific type of protein, hexafoliatin. Okay, so these are the various type of Staphylococcus skin infections caused by the Staphylococcus aureus. Now, along with these staphylococcal infections, Staphylococci also produces another type of protein. That protein we call as enterotoxin A. And this enterotoxin A, it causes a form of food poisoning. Okay, so this enterotoxin A, it is responsible for causing a kind of food poisoning in the person, which that person ingested the suppose this toxin is present in the food, and such contaminated food is taken by a healthy person. Then this toxin stimulates T cells localized along the intestine, resulting in massive T cell response and release of inflammatory mediators. And the final outcome is the severe short-lived diarrhea and vomiting, which is associated with the staphylococcal food poisoning. So this staphylococcus produces two types of infections. First is the staphylococcal infection, and the second is the intoxication. And this intoxication, food intoxication, it is produced by the staphylococcal enterotoxin A. So this is all about the pathogenesis and symptomology. Then the laboratory diagnosis. Now, in order to diagnose this disease, the samples, the specimens which is to be collected depends upon the type of lesions. कोणता lesions तो दाखवत आहे त्यावरून आपल्याला त्याचे सॅम्पल स्पेसिमन सॅम्पल कलेक्ट करावं लागेल. Now, if superiority lesions are there, then we can collect pus samples. If respiratory infection is there, then we can collect sputum sample. So depending upon the type of lesion, we can select the source. Now in case of food poisoning, feces and the remaining suspected food should be used as a sample for the source of this. For the detection of the carriers, the nasal swab is used. So usual specimen for the detection of the carrier is the nasal swab. Swabs from perineum, species of the hairs, umbilical stumps may be necessary for special conditions or special situations. Now, what we perform then? If we have taken the sample from the pus from the superiority lesions, then direct microscopy with gram staining is useful in the case of your pus. When we observe the pus by using the gram staining, we are able to observe grape-like clustered gram-positive cocci. This is the characteristic of this bacteria. This is of no value for specimen like sputum. Sputum sample जर collect केला असेल तर sputum sample हे useful नाही because that sputum sample contains mixed bacterial flora which is normally present there in that sputum sample. Now diagnosis also carried out by using cultural methods. The specimen which we have collected directly plated on the blood agar, and the characteristic colonies which are produced by this bacteria on blood agar shows the various kinds of hemolysis, alpha, beta and gamma type of hemolysis. Here on this blood agar, these colonies they appear after overnight incubation. Now specimens were collected where staphylococcus are expected, they are to be scanty and outnumbered by other bacteria. So we have to use specific selective mediums such as Ludens, salt milled agar, Robertson's cooked meat medium which is containing 10% sodium chloride. So such specimens which is having less number of bacteria, less number of Staphylococcus aureus bacteria, then we have to plate that or spread that back specimen sample on the selective medium. So we are able to get the large amount of colonies on these selective mediums. And after that we have to perform a specific test which is most commonly used for the identification of this Staphylococcus aureus bacteria. That is the coagulase test. Now the coagulase test, it is carried out by two different ways: by tube agglutination test, tube coagulase test, or by slide coagulase test. In tube coagulase test, it is used for the detection of free coagulase. Here 0.1 ml of young bacterial culture or agar culture suspension of the isolated bacteria is taken, and to that 0.5 ml of human or rabbit plasma in a narrow test tube is added. So in tube coagulase test, it is used for the detection of free coagulase enzyme. For that purpose, we have to take the 0.1 ml of bacterial suspension, which we have to add in a 0.5 5 ml of human or rabbit plasma in a narrow test tube. Here we can use the EDTA of oxalate or heparin may be used as anticoagulant for preparing the plasma. Now citrate it is not recommended because it may be utilized by some of contaminated bacteria causing false positive test. So here positive and negative controls are also set up. These tubes they are incubated in a water bath at 37 degrees Celsius temperature for 3 to 6 hours. If positive, the plasma clots and it doesn't flow when the test tube is tilted. And continued incubation is not recommended as the clot may be get lysed by fibrinogen formed by some strains. This is the coagulase test which is done in a test. Similarly, coagulase test can be done on the slide, which can be used for the detection of bound coagulase. And here, for the slide test, the isolated bacteria it is emulsified with saline on a slide. After checking the absence of autoagglutination, a drop of human or rabbit plasma is added there, and then it is emulsified and mixed. If prompt clumping of cocci indicates the positive test. Here also positive and negative controls are also maintained. Then the isolated culture should be tested for antibiotic sensitivity test. So by doing this antibiotic sensitivity test, we are able to detect whether this bacteria is sensitive to which kind of antibiotics. And this is also helpful because this also helps us in the treatments also. Bacteriophage typing may be done if the information is desired for epidemiological purposes. Other typing method includes antibiogram pattern, plasmid profile, DNA fingerprinting, ribotyping, PCR-based analysis for the genome pleomorphism. These are the other methods that can be utilized for the diagnosis purposes. Then the prophylaxis of this staphylococcal infection. Prevention of staphylococcal infection is problematic because most individuals which carry this pathogen, they are the asymptomatic carriers. And diseases such as SN and impetigo can be transmitted by simple contact with contaminated fingers. So it is risky one. So prevention is risky because of these factors. Now this prevention strategy includes exclusion of infected individuals, including carriers, and elimination of pathogen. Now in hospital environment such as surgical wards, nurseries, carriers of the known pathogenic strain must be either excluded or treated with topical or systematic antimicrobial drugs to eliminate the pathogens. If in a hospital staff some carriers may be detected, that may be separated and that should be treated in order to avoid the spread of this infection in the hospital area. Now in one study, the nose of the patient were swabbed and cultured before surgical procedures. The patient who were positive for Staphylococcus aureus were treated intranasally with topical antibiotic, a body wash of chlorhexidine, or topical antiseptic. So depending upon before any kind of surgery, that person is tested for the presence of any kind of staphylococcal infection. If that person is suffering from any kind of staphylococcal infection, then that person firstly treated for this infection and then the surgery carried out. Then finally, if that disease occurred because of the staphylococcus, then how to treat that includes the treatment. Drug resistance, it is most common among the staphylococci. So appropriate antibiotic should be chosen by using the antibiotic sensitivity test, which we have carried out in the laboratory diagnosis. This is useful for the treatment purpose. Now, benzyl penicillin is the most effective antibiotic if the strain is the sensitive one. नॉर्मल पेनिसिलिन हे खूप त्या पेनिसिलिनला काही स्ट्रेन सेन्सिटिव्ह आहे पण जे रेजिस्टंट झालेले आहेत सो in such cases we have to use another kinds of antibiotic. Methicillin was the first compound developed to combat resistance due to penicillins production by Staphylococcus. This is another kind of antibiotic which is used in those patients where the Staphylococcus is resistant to the antibiotic penicillin. Okay. Now, due to the limitations of the clinical use of methicillin, cloxacillin are used instead against the penicillin producing strains. So, those Staphylococcus aureus strains which are resistant to penicillin, in such cases, cloxacillin, this is another kind of methicillin antibiotic that should be used. But the methicillin resistant strains of the Staphylococci become common, which are resistant not merely to the penicillin but also to other beta-lactam antibiotics and many other kind of antibiotics. So, in such case, other antibiotics should be used to treat this disease. Now, in life-threatening conditions of staphylococcal infections, vancomycin is the drug of choice. If the strain is resistant to the vancomycin and teicoplanin have to be appeared in the hospitals where the antibiotic use of this antibiotic is indiscriminate use. In some hospitals where these antibiotics they have been used in tremendous amount, in such cases this bacteria also became resistant to this vancomycin and teicoplanin and antibiotics. For the mild superficial lesions, systematic antibiotic may not be necessary. Topical applications of drugs not used for systematically. Okay, bacitracin, chlorhexidine, mupirocin, they may be sufficient for the mild superficial lesions caused by the Staphylococcus aureus. Now, some strains of the Staphylococcus aureus, they also show another kind of phenomena that is known as a drug tolerance. These strains will become found to be susceptible in the sensitivity test. When we test that Staphylococcus aureus strains in laboratory for antibiotic sensitivity test, these strains they are found sensitive there, but their minimum bactericidal concentration will be very much higher than the minimum inhibitory concentration. The amount of antibiotic which is required is very high in such conditions. They are not killed by antibiotic in usual doses and persist there, leading to the failure in the eradication of the infections. So this drug tolerance, it is another kind of phenomena which is shown by the Staphylococcus aureus species or strains, because in laboratory we found these bacteria they are sensitive for the antibiotic for lower concentration, but while in treating the patient at that time this much amount of concentration of antibiotic it is not sufficient, but the higher amount of antibiotic is required there. So this is known as a drug tolerance phenomenon. And here for the treatment of carriers, local application of suitable antibiotics such as bacitracin and antiseptics such as chlorhexidine, they are sufficient. In resident cases, they causing many problems. And in such condition, rifamycin, which is given orally, is effective for long-term suppression of and elimination of the carrier steps. So, for the treatment of carriers, bacitracin and antiseptic chlorhexidine used. But if that strain is resistant to these antibiotics, then another antibiotic, rifamycin, it is used, which is given orally, and so that's why this bacteria it is killed from that carrier, and that carrier becomes healthy now. health, then becomes health.